So today we're going to talk about meiosis. There are a lot of similarities between meiosis and mitosis. However, they have very different purposes and they have very different outcomes that suit those purposes. So if you haven't had a chance to actually copy down those different phases of mitosis and what happens in them, make sure you do that now before you proceed, because I'm going to be focusing on the differences in meiosis rather than just trying to go through all of the similarities as well. Take a moment to read through the learning goals and copy them down in your learning log, and then press play when you're ready to continue. When we're talking about meiosis, we're talking about the formation of gametes. So sperm in men and egg in women. Mitosis was talking about the formation of somatic cells. So that is every other cell in your body, your hair, your skin, your brain cells, all of those other cells are somatic. And that's because they all contain the exact same DNA. One set from mom and one set from dad. Gametes are a little bit different. They're what we call haploid cells. Haploid cells, you can think of it like hap as in half. They actually have half the amount of DNA, or only one set. So when you see that N, think about it talking about a set of chromosomes. Most of our cells are going to be diploid, that di meaning two. And we actually have two sets of DNA in the rest of our body. If I looked at my skin cell, I'd be able to see DNA from both my mom and my dad. So I have two different sets of chromosomes, one from each parent. In haploid cells, we don't have that. We only have one set of chromosomes. So at that point, it has half the amount of DNA as anything else. We'll get into why that happens, but it connects really well to the purposes of meiosis. One is talking about the fact that we need genetic diversity. If you happen to look at your parents, you probably don't look exactly like them. So something must happen during reproduction and the creation of those gametes to allow for some difference. The other thing is that we need to be able to half the amount of DNA that we have in our sex cells. If we happen to have 46 and 46, those would combine and create a child with 92 chromosomes. It's way too many chromosomes. Have so many disorders and so many issues with the way that their body worked. So in the same way in mitosis that we have DNA replicated, before we get into meiosis, we still have that duplication of DNA. Meiosis actually consists of two stages. In mitosis, we had 4 phases: prophase, metaphase, anaphase, and then telophase. And we finished that off with cytokinesis. In meiosis, we actually go through that twice. So we have 2 prophases, 2 metaphases, 2 anaphases, and 2 telophases. And those are really important to have all of them in order to accomplish what we want. So in the first stage, as we're going through, we're looking at homologous chromosomes being separated. In the second stage, when we go through, we're actually looking at those sister chromatids being separated. And that's where we get our half of the DNA that we would normally find in all of our other cells. Because we have those chromatids being separated, we're going to actually have 4 cells at the end instead of the 2 that we get from mitosis, and all 4 of those cells are going to have half the amount of DNA. We'll take a further look at that one. So when we're looking at what's happening with chromosome numbers, here's a nice little picture to hopefully clarify some of the things. I just want to point at this one. So we've talked about this a little bit already, how chromosomes are like sets of DNA. Here, even though those chromatids are connected to each other, that still counts as a set. Remember, this one is a duplicate of this one. And so because they're physically connected and it's a set of DNA, we actually have 46 chromosomes, even though we have double the amount of chromatids. In the next one, because that gets halved, we get 23 chromosomes and 46 chromatids. And then that last one there, when we separate it out for meiosis, we actually have 23 chromosomes and 23 chromatids. So if you're still confused about chromosome versus chromatids, remember, when they're physically connected together at that centromere, that's still one segment, one set of DNA. And so we count that as one chromosome, even though it might actually be double the amount of chromatids. Let's take a look at see how this actually comes about when we're looking at things like sperm and egg. And I'm going to introduce a couple words to you. So let's take a look to see what's actually happening when we create sperm and egg. Going through the process of creating gametes is something called gametogenesis. Genesis means creation, and so that would just say the creation of gametes. If we're looking at what's happening in the father, this is where we come into our DNA would have been created. So here we've got 46 chromosomes, but we actually have 92 chromatids. When that separates, we get 46 chromatids in each one. And then when those ones separate again into two, then we're going to have 23 chromatids in each of those gametes. So keep in mind the difference between chromosome and chromatid. For the father, since it's creating sperm, we call that spermatogenesis. And you'll notice that we have one cell and we actually get 4 sperm cells. Something a little bit different happens when it comes to females. Females, since we're creating ovums, which is another word for egg, it's called oogenesis. And in women, since eggs are so much larger than sperm, we actually take a lot of the cytoplasm and so on from these other cells to create that one egg. So for females, we actually only get one egg from one cell, but we've gone through some very important changes along the way in order to cut that DNA down to half the amount and also to create some variety. So even though we look at, a lot of times when we're looking at meiosis, you're going to see 4 cells created from one, and that's generally what we talk about. We have 4 cells here, but only one's actually functional. So one thing to note that is different when we're talking about going from meiosis 1 to meiosis 2 is we don't have any duplication in DNA. And that's really important because if we duplicated that DNA, then we wouldn't be able to half it at all. So we make sure that no DNA is getting duplicated so that when we go through the division again, we're actually cutting down the amount of DNA in each cell by half. Let's take a look at the general stages that happen. You'll notice a lot of the same similarities. Prophase, you have that formation of chromosomes, you have the nuclear envelope dissolving, and you have those centrioles that are moving towards different sides of the cells. So metaphase, homologous chromosomes line up, and so homologous pairs are pulled apart from each other. So here we've got one pair, and red always means mother for some reason, so we'll say the red is the mother, the blue is the father. We've got one pair of the red one going to one side, one pair of the blue going to the other side. So that's separation. When we're looking at meiosis 2, we're looking at what happens with those chromatids. So because there's no duplication, we have those pairs of homologous chromosomes that move to different sides. They now move towards the center of the cell, and those homologous chromosomes get broken apart. It's the sister chromatids that separate and go to different halves during anaphase. So big difference, anaphase 1 and anaphase 2 is what's going to be separating there. Just a little bit of an overview looking at the differences between mitosis, meiosis. So synapsis is actually talking about the process that happens when we get 2 homologous chromosomes coming together, and they form a tetrad. So if you remember what tetra means in things like chemistry, tetra equals 4. So a tetrad means that we actually have, these are our homologous chromosomes, they come together, and now we have 4 chromatids that are physically connected with each other. Crossing over happens when parts of the DNA actually, or sections of the DNA actually cross over with each other, and they're going to switch different sections of genes. So here, we happen to have these little bits get switched, but those sections code for the same gene, so the fact that they're getting switched just adds more variety. And now we've got, in the end, this one, instead of being all blue like its neighbor, it's got different chunks of DNA. And this one, instead of being all red, has some of that blue one. So if red is maternal, this sperm might have only maternal DNA, and this would be the father's mother. And this cell has a mix of maternal and paternal DNA. We talk about this as recombination because we have recombined genes together. What gets created is no longer the same as what existed before. Something else that creates some of that variety in meiosis is something known as random assortment. You might also see it talked about as independent assortment. They're the same things. That just means that when we have chromosomes lining up during metaphase, the way that they line up is completely random. We might have these two chromosomes line up, and so when they split, it makes sense you'd have one half go to one side and one half go to the other. But perhaps we have things different. And in that earlier stage, we had a different second cell that was created. Now we've got some paternal DNA and maternal DNA in the same cell. So that when we separate, these actually have a combination of both. So obviously, here we're just looking at two different chromosomes. We have 23 that's happening in the cell at each time. So we're going to have lots of different ways that we can combine maternal chromosomes and paternal chromosomes so that we'll have like a lot of variety as to what options are there. In order for us to make sure that we keep the numbers down, and we're not going to have some offspring that has double the amount of DNA, because that would be a lot of difficulties and probably a lot of disorders, we need to have some kind of process that separates these out. This is known as the law of segregation. And that's simply talking about we have two chromatids connected together in that chromosome. We have to separate them out to be able to get half that DNA. It means that when we have sperm and egg come together, we have N, one set of DNA, maybe from mom or your mom's mom, and another N, another set of DNA, your mom's dad, which come together to give us that full set of 2N. Or you can think about it as in we separate out, so we have 23 chromosomes plus 23 chromosomes, one from sperm, one from egg, and they give us back our full 46 chromosomes that we need to function properly. Take a look at these key questions. We'll come back to them tomorrow and spend most of our time focusing on these, but go through them and reflect on how well you understood this lesson. And bring any questions you might have to class tomorrow.